Hey guys, welcome to our lab featuring Jenna, Stephanie, Eric, and Sarah. Don't forget me, Aiden D. The purpose of this experiment is to see how peeled eggs react to multiple solutions in regards to osmosis and, what's the other one?
Diffusion.
Diffusion. We can apply this knowledge to the cell membrane. What's our hypothesis, guys? Our material list starts here. 3 cups.
3 eggs held in vinegar for 24 hours.
284 milligrams of table salt.
200 milliliters of table syrup.
400 milliliters of distilled water.
Safety glasses.
A scale.
100 milliliter graduate cylinder.
2 drops of food coloring. 300 milliliter beaker.
Okay, so the first thing we did was mass our eggs.
Shield took our scale, so
So we have each of our eggs in 3 different cups. So we will fill up each cup with 200 milliliters of different solutions. Our first solution will be this corn syrup over here. Our second solution will be 100% distilled water. Our third solution will be sodium water.
Right now I'm gonna put the 200 milliliters of corn syrup into this blue cup labeled maple syrup. It's not actually maple syrup. Then you put the egg inside of it. Okay.
okay, I'll grab some eggs.
I honestly don't think the egg will.
Wait, who took our scale? Oh, Shield took our
Make sure the rest of it is in there.
Now we're just gonna plop in the egg. Plop.
You're gonna be in here, you understand that, right?
If the egg is placed in a maple syrup solution for 24 hours, then the egg will decrease in size because the maple syrup solution is hypertonic.
We are now mixing the food coloring with the distilled water to create a rose-colored distilled water solution. That looks good.
Right here I have 200 milliliters of distilled water that I dropped 2 things of red food coloring into. Now it's red distilled water. Now I'm going to pour the 200 milliliters into this blue cup that is labeled as so. Distilled water. Now we are going to place in the second egg.
Plop.
That's all, folks.
In our solution with the distilled water and the egg, our hypothesis is if the egg is placed in distilled water, then the egg will become larger because the egg has a greater concentration than the distilled water. So due to osmosis, the water will go into the egg because the solution is hypotonic. Right now we are creating our table salt solution. I'm adding 2.84 grams of table salt to 200 milliliters of distilled water. This should be the same ratio of sodium to water as there is in the egg. That will create an isotonic solution. That's the hope. We will see tomorrow.
So now we have our table salt and water solution, and it's already mixed up, and we're gonna pour the 200 milliliters into our salt cup. It's labeled salt.
Now we're going to add the egg. Plop.
Our second hypothesis is if we place the egg inside of our saltwater solution, and that solution has the same or similar concentration to that of the egg, then it will be an isotonic system, and the egg will hopefully not shrink or grow.
No, you do it.
This is before we leave for the night. Distilled water, maple syrup, corn syrup, and salt solution. Can't wait to see how our eggs fare in the morning.
After a night of rest, we're back 24 hours later to look at our eggs. So here in the distilled water solution, you can see that our egg has grown a little bit. Here in our salt water solution, you can see that the egg looks pretty much the same. And then in our last corn syrup or maple syrup solution, you can see that the egg has shrunk a lot. Let's measure the masses to see just how much it has grown or shrunk. Our initial mass for the egg in the table syrup solution was 93.26 grams. Now, after 24 hours in the table syrup solution, it is now 54.229 grams. Outstanding. As you can see, it's super tiny. And if you look in the table syrup solution, you can see it's kind of watery. That means that water did escape from the egg. As you can see, our initial mass of the distilled water egg in the distilled water solution was 90.63 grams. Now, over 24 hours in the distilled water solution, our egg is now 93.71 grams and has increased in mass.
You can also note that the egg is now red, indicating the water that was dyed red from the red food coloring has seeped into it.
So our initial mass of the egg in the saltwater solution was 90.59 grams. After 24 hours in the saltwater solution, it is now 93.10 grams. So it did grow, contrary to our hypothesis that it wasn't going to grow, but it didn't grow too much.
What this tells us is that our concentration of salt was a little bit too less when compared to the concentration of the egg. This means the solution was not isotonic, but it was in fact hypotonic. Our conclusion is this. Although we were correct in our hypothesis about the table syrup and the distilled water, we were a little bit off by saying that the egg in the salt solution would stay the same. It actually grew just like the distilled water.
Our first source of error was that all of our eggs were not the identical mass.
Our next source of error is that the eggs were not all soaked in their solution for exactly 24 hours each.
We failed to measure one of our solutions in a graduated cylinder. Instead, we used a beaker.
Another source of error is that the amount of salt we put into the salt solution, which we had hoped to be the same concentration of the amount of salt within the egg, was not correct.
Hey guys, Aiden here. I was wondering to myself, why is it that the water in our saltwater solution moved into the egg even though we thought we had the same concentration of salt on the inside of the egg than there is on the outside of the egg? I realized that perhaps there's a different compound inside of the egg that is simply not present in our saltwater solution. This could be why the water moved inside of the egg because, well, this compound has a relatively high concentration relative to the outside of the egg, where there could be practically no concentration.
Here you can see simple diffusion. In simple diffusion, small uncharged polar molecules and hydrophobic molecules like CO2 and oxygen gas move directly through the phospholipid bilayer of a cell membrane. The movement of the substance is from an area of high concentration to an area of low concentration. Each substrate diffuses down its own concentration gradient, unaffected by the concentration differences of other substances. There's also facilitated diffusion. This is the diffusion of polar molecules and ions across the membrane with the help of transport proteins that span the membrane. Each transport protein is specific for the solute it transports. This process requires no energy input to move the substance.
Osmosis is the movement of water across a semi-permeable cell membrane. The water starts at a hypotonic solution that has a low concentration of solute. It moves across the cell membrane to an area of high concentration of solute called a hypertonic solution. Here, it creates an equilibrium called an isotonic solution. The net movement of water across the membrane when it is at equilibrium is 0.
In diffusion, the particles move from high to low concentration of solute, and in osmosis, the water particles move from high to low concentration of water. They are both passive, meaning they require no energy. They both move through the cell membrane, and they can both use proteins. Diffusion is the movement of solutes, whereas osmosis is the movement of water.